simply by that. Okay. So what calcium does? I'm just gonna go, quickly this. Calcium activates probably. I think if you go into the literature, you will say there are probably like 200 molecules that people claim that the calcium activator is good for ATP. Okay, so forget about that. So, and everybody will claim that their molecule is the most important, but everybody, the most common thing, it will be this molecule called chemkinase 2. And this is a protein kinase that is particularly activated by calcium. Okay? And one of this is the most common effector of calcium, and the experiment, that's pretty bad. Here I explained the whole experiment, and I guess I lost it. All right. Anyway, I'm going to have to explain it with my words. So in this experiment, what they do is this enzyme, chemkinase, you activate it with calcium. And remember the NMDA that calcium in. So basically the idea will be that calcium activated chemkinase. How you can demonstrate that? Well, what you do, if you take a flask full of chemkinase, put some calcium in there, activate it, and then put it in the little pipette that you're using to record from the cell. So now the activated chemkinase is going to flow inside the cell. And if the consequence of an NMDA receptor is the activation of the chemkinase, if you're providing chemkinase, active chemkinase by yourself, then you don't need to do the tetanus, you don't need to do anything. And basically this is shown here. So what you have here you penetrate into the cell, and here you can see that in this experiment where you have this pipette for recording with the chemkinase active in there, you start to get a bigger response. Okay. Not shown in here, at that point the guys tried to make a tetanus and nothing happened because tetanus LTP was already induced. Now in this one, what they have done here is they take the same chemkinase, but they boil it a little bit to inactivate. And in that case, nothing happened. But when you go in here and you try to use the tetanus, then you do the tetanus, then you use LTP because LTP never happens. Okay? You follow that? I see that. You got it? Okay. I take it your word. All right. So the important thing is that if you put a good activated chemkinase, you mimic LTP basically, and you can bypass the NMDA receptor, and you bypass all of those things. Okay. So the expression, I think we're going to finish here because. So what did change? So I activated the NMDA receptors, but now I'm going to make the synapse stronger. How I can make the synapse stronger? And here are the possibilities. On the presynaptic side, I tell you that you release a vesicle with the neurotransmitter, so you can do two things. Either you can, in each one of these vesicles, you can put twice as much as a neurotransmitter, and then you're going to get twice as much of a response, more or less. Some non-linearity is involved. Or you can increase the probability of release. I don't know if you are familiar with the synaptic transmission, but actually anytime you get an action potential at the end of the terminal, not always you release a vesicle. You release with a probability of release. Okay, a probability which is not 1. Very often it's kind of very low. Actually, these cells in the hippocampus, these synapses in the hippocampus that I've been telling you, the probability that they release a vesicle upon an action potential is very low, it's 0.1. 1 out of 10 times they will release the vesicle. So if you increase that probability of release, you're going to get a bigger response on average, because normally you're recording for several division of several axons. Okay, that will be one. Now, but postsynaptically, you can have more receptors. You can have more of those AMPA receptors. If you put twice AMPA receptors, you get twice the response easily. Or you make it, change the properties of the receptor. You make it, they open bigger, they open more often, all kind of things. So this actually was one of the most interesting times of synaptic plasticity fields in the 90s. because the field was bitterly divided between the presynaptic guys and the postsynaptic guys. And you will go to the conference and those guys say, you cannot say that because you are... And then the most, and I was a kid in there, but it was kind of fun to watch all of those guys fighting. All right? So the camp was divided between the presynaptic side and the postsynaptic side until the presynaptic guys make an incredible creative experiment that switches the whole thing to the presynaptic side. And this is a good story. So, as I told you, synaptic release is a probabilistic phenomenon. So sometimes you get it and sometimes you don't. Okay? So here what we have, this is what is called with a single axon stimulation. So here you do one single axon and you are seeing the effect of that. So sometimes, as I told you, it's a probabilistic thing. Sometimes you don't release anything. Sometimes you release one vesicle. Sometimes you release two vesicles, and sometimes you release three vesicles. So you can make here a histogram, failures, one vesicle, and two vesicles, and three vesicles. You can see that, all right? Now, and very often it's failed, okay? Most of the time it's failed. So this is the number of cases that you get a failure. So now, they took that diagram, and then they do LTP. And what the guys found, it was brutally that you get more vesicles, but actually a fewer failures. So you can comment or you can discuss, whether you have more vesicles or not, but the reduction of the probability of failures is an increase in the probability of release. Okay, you will follow that. Okay, so the fact that they are getting less failures, it means that you have more release, and that for a while it settled the thing. This thing is just you are controlling the probability of release. Right? Now, that was part of the story. The next part is very cool because it was totally the opposite. And actually, the field of plasticity I think have been driven basically by people with bright ideas. Okay? And as I told you, here have these bright, insightful ideas, and this is another insightful idea that it was advanced by Francis Edwards. Actually, it's a bright woman. At the beginning, I thought it was a man. That showed my misogyny. Okay, but it's a bright woman, and this idea is beautiful. And it's the following. So she said, okay, so we have a failure, but just because we have a failure, it doesn't mean it's a presynaptic failure. It's not a failure of release. And here's the creative thinking. She said, well, most of the experiments that people do, they do it in very young tissue because it's easy to manipulate and it's easy to record. And in very, so that's one thing. And the other thing is like all of those experiments you maintain for simplicity of the recordings, the voltage of the cell at a very negative, voltage. Okay? And she said, well, what happened if the synapses, when they're born, they only contain an NMDA receptor and no AMPA receptor. Okay? So what will happen? Well, if you're going to see this one and you're keeping this synapse at a very negative voltage, you're not going to see anything because remember that the NMDA receptor is normally clogged by the magnesium ion in there. So if you record from a synapse that only contain NMDA receptors and you're maintaining the cell at a very negative state, you're not going to see anything. You're going to say that's a failure, there's no synapse in there. Now, suppose that what happened is that after LTP, after inducing LTP, what you're getting is that you're getting the insertion now somehow of AMPA receptor. Okay? Now you're going to see a response. So you go from a situation in which you have no response first, and now you have a response, but it's not because it's a change in the probability of releasing vesicles, but it's basically before you didn't have any AMPA receptors, but now you have it. You follow that? Okay, and they call it that silent synapse. Okay, and that was a. Huh?
How does the younger nerve with only the FDA form, or like get to a positive enough voltage to form?
Excellent question, beautiful. So it turned out there are many. So first of all, you have that noise. In all of these systems, you have noise, and you can have that. But interestingly, when the animal is very young, synaptic inhibition provided by GABAergic receptors, it's involved the chloride crossing the membrane, is actually depolarizing, is excitatory. So in the very young guys, in the rats, for instance, postnatally, GABA, there is no inhibition basically. Inhibitory synapses are actually excitatory. Okay. And then what happened, because all of these synaptic changes depend on the gradients. Okay. If you have more of the iron in there, like here, the iron will go this way and the other way around. But what happened is that when the animal is very young, the gradient of the chloride is the opposite as in the adults. Okay. And all of that is because you have different set of transporters. Okay. But basically, in the young guys, inhibition is going to help excitation. And once everything is done, basically inhibition will go to inhibitors. That's it. This is a beautiful chapter of neuroscience, but I have to talk now only about this part. Okay, so evidence for that. Only 2 minutes. Let me see. Okay, yeah, I'm gonna go for the evidence. So this is the first. We're gonna go to this one. This is the first demonstration that actually you can have a synapse without receptors, and then after ATP have AMPA receptors. What they have in here is the culture cells, okay, and they infected with a virus that forced the cell to produce a lot of AMPA receptors with a tag, with a GFP, a fluorescence. Okay, so you can illuminate with one wavelength and get the excitation back, and then you can see whether the signal is. So basically, this is the dendrite full of this, so to speak, full of this AMPA receptors with a fluorescent tag. Okay, and what happened, and you don't have to follow this part. What they do is they do LTP in this moment. This is before and this is after, and you can see that in A there was nothing here, and now you have that spine full of AMPA receptors. So there was a spine in here that it was empty of AMPA receptors, and now it was full of AMPA receptors. Okay, and that's basically a silent synapse that became non-silent. Okay, this guy didn't have any, and now it has. So now that was done in the late 90s. Okay, and that was kind of fast. The technology now have advanced, and now this is a state of the art. The guys are showing basically the same thing, okay, but not in culture cells, but in a live animal behaving, and then they're stimulating our whiskers, and then you can see that stimulating the whiskers of the rat, and then you can produce, and these little arrows, you can get more from here to here, you get more AMPA receptors, okay? And these are AMPA receptors that only fluoresce when they're outside, expressed outside of the membrane. So it's basically a lot of technological improvements are going, have happened since then. But basically, it's the basic same principle, okay? So the guys make a very beautiful conclusion with a bad images, and now we're confirming with beautiful images. So, and since we are about to finish, because is that 4:20? Yeah. Okay. So this is basically the model that people have, is that activation of the NMDA receptors produce calcium, and the calcium activates CAM kinase, and somehow it enforced the movement of AMPA receptors that were kind of outside of the synapse to get into the synapse and get trapped and anchored in there. Okay. So these guys, it was kind of like on the side of the synapse, and now it goes in here. And that's RTP. Okay. This little arrow in here. All right. So and how can kinase do that? Nobody knows. Okay, I mean, or there are like 25,000 different theories about or experiments and stuff like that. But basically, the fact is that after LTP, the receptors that were kind of like diffusely around here, they get inserted into there. And I was supposed to talk about LTD, which is the other way around, but basically, I'm gonna skip everything except I'm gonna tell you that LTD, the depression, it also requires an NMDA receptors. And people were saying, whoa, why is that? You know, to put the AMPA receptors, you require an NMDA receptors. To take them out, you also require an NMDA receptor. How? Explain that to me, okay? And the explanation is simple, okay? So basically, the idea is the amount of NMDA receptor activation. If you activate a lot of them, you get a lot of calcium, and you get a lot of this chem kinase, all right? But if you don't activate a lot, right, kind of like a little bit, all right, you activate basically phosphatases, and those phosphatases undo the work of the chem kinase. Okay, so the kinases are good for putting the receptors in the synapse, and the phosphatases are good for take them out. And both of them are commanded by NMDA receptors. In one case, you need a lot of calcium to do it, and the other one you need a little bit of calcium to do it. But basically, that's the same. All right? Yeah, I am 3 minutes over, so. Yeah? Good. My understanding is until 2:20, is that true?
Yeah, it's 2.
And it's 2, huh?
It's 2:15.
2:15? Well, this guy here says it's 2:23. Sorry, guys. Okay, so, well, Jesus. It's the computer. Okay. So, all right, so then I'm gonna tell you a little bit more about hippocampal LTD because it's the other side of the equation. And there is a. Anyway. So, not only, and there was a good, people start to look for LTD after the successful elucidation of LTP. And there's 2 good reasons, okay, or actually 1 good reason. You cannot have just LTP. You cannot just potentiate the synapses. I mean, eventually, you're going to saturate them. I mean, you cannot cram, 10,000 AMPA receptors in a poor spine, and the thing is going to explode, all right? And the idea is that what you have anytime you learn something, it's not just more strength of synapses, it's just redistribution. And you can see it easily if you take a rat and teach them, okay, in a classical paradigm like a Pavlovian, go to the left because in the left you get the water. And the guy goes in there and get a TP to get the water in there. And then you change the paradigm and say, no, the water now is in the right. Okay? So now the rat had to do LTP for the other part of the other synapses, meaning going to the right, but also have to do LTD and do the previous learning. Okay? So anytime, very often when you're switching preferences, it means 2 things. Potentiate the new preference and depress the older preference. So you need LTD. You need both LTP and LTD. So people were looking for those. And here is how you get normally in the lab. So people were looking, and the question was which would be the paradigm for getting it? And this is a beautiful thing because I was there when LTD was discovered. So I was a postdoc and the graduate student who discovered that. She was trying to do different paradigms, different frequencies and stuff like that. And at some point say, okay, I'm gonna do a very short stimulation with 1 hertz. Okay, so we need a high frequency stimulation for LTP, 100 hertz. Let me try short frequency stimulation for a few seconds, not that much. And then, but it was the day when the speaker came to give seminar, and it was the pizza lunch. So she went there, and then she started to eat the pizza and forgot that she was setting the low frequency stimulation. And when she came back, oh crap, my experiment. She came back, like 20 minutes had passed, and they would give her 1 hertz stimulation all the time, and say, ooh, and then she found the whole thing depressed. And then she repeated, and it turned out that actually one prolonged low frequency stimulation did effect. Okay? And then with that, the first question she asked, it was, well, if NMDA receptors are so good for LTP, well, if I give you 1 Hz stimulation, low frequency stimulation, and give a blocker of LTP, I should get even more LTD, more depression. I'm going to block the NMDA receptor then. And she blocked the NMDA receptors, and actually she couldn't get any LTD. So that was a weird stuff. You know, you're expecting to have more LTD because you're eliminating the potentiation, and now you get nothing. Okay? And then that was when people start to work out on the mechanism, and it turned out actually that is the difference of effectors. Okay? So the Kim kinase, you require a lot of activity, a lot of calcium for them. And the cool thing is that if you don't do anything, nothing happens. Okay? If you activate a little bit of the NMDA receptor, a little bit of calcium, you get the phosphatase activity. And then with more, you get the Kim kinase activity, but the Kim kinase beat the phosphatase. So at that point you have both Kim kinase and phosphatase. But the Kim kinase is much more active, so it overrides the phosphatase. And that's the way that you get that. Right? And I think, yep. And there's other type of LTPs and LTD that you don't care. Okay. Next time we are going to talk about how you use LTP probably in real life. Okay, how that happens in the brain. And I'm going to talk about the other things. And now it should be like about 4:20. What time is it? Oh, perfect. Yeah. So I see a few more people in here. So my name is Alfredo Kirkwood, and I'm going to give the lecture today. And basically, last time, I tried to show you how wonderful long-term potentiation is as a model for learning and acquisition of new memories. And today's lecture is going to be to show how inaccurate and incomplete that was, okay? So inaccurate because actually the ways that we induce LTP and LTD in the lab are very likely not the way that happen in the real world. And two, if, I'm gonna show you that too, that probably if you have a system that is only endowed with a long-term potentiation and long-term depression, it's very likely it's incapable of learning anything, okay? So you need something else. So, and those are going to be, basically I'm going to talk about spike time independent plasticity and the regulation of LTP and LTD. So let's start with the beginning. And I'm just going to remind you the current model of LTP and LTD. So everything is about the NMDA receptor with other forms, but we're not talking about them today. And if you have NMDA receptors get open when there's pre and post synaptic activity, and calcium gets in, and if you have a large surge of calcium, like during a tetanus, you activate kinases and you get LTP. And if it's not that big, you get phosphatase and you get LTD. Well, and actually that is true. And the ways of people are showing is that you can vary the amount of calcium inside the cell or the amount of NMDA activation, and you can go smoothly from LTD to LTP. So one way, for instance, is shown in here. So what we have in here is that we activate we stimulate the synapses at a given frequency, and we change the potential of the membrane of the cell to different levels. So if nothing happened, normally the cell is at minus 60 or 80 here, nothing happened. But if you change the potential a little bit, kind of like minus 40, then you get depression. And if you increase it, you get potentiation. So the idea is that at this level, the NMDA receptors are kind of active, but not so fully active. And here, they're totally active. And the same thing with the frequencies. If you stimulate a low frequency, you activate a little bit the NMDA receptors. And if you stimulate at higher frequencies during the tetanus, you activate them fully. But those are not good how you say, paradigms of how it happened. Because in order to get LTP here, you need high frequency stimulation, high frequency like epilepsy. And you don't need epilepsy for learning. And here, to get depression, you need 1 hertz. And you never get 1 hertz stimulation constantly unless you go to a bad disco and you're gonna boomba, boomba, boomba. Okay, but that it doesn't happen. Or you have to depolarize the cell to a given level for several minutes. That also is not happening unless you get a knockout or something like that. So they're totally unrealistic ways of inducing plasticity, but they're effective in the lab to study the mechanism. Okay, so it's an effective way, but unrealistic way at the same time. So if we go to the initial thoughts of Hebb and other people, what you need is the correlation. So here you have a pyramidal cell in the brain, and here you have input from one side and input from the other side, and this is the output. And the idea in here is like, for instance, this is trying to recapitulate an older experiment by these guys, Hubel and Bissell. Hubel and Bissell in the 60s, what they do is they close one eye when the animal is very young, and after closing for a few days, it turned out that the eye gets disconnected from the cortex. The cortex doesn't respond to that eye. And the idea is the following. So normally, the two eyes, the left and the right eye, are firing action potentials. These things in here will be action potentials. And since you're seeing kind of like the same thing with the two eyes, normally, okay, you make this postsynaptic cell to fire action potentials kind of like in the same, at the same time, and everybody's correlated, and that's good. But when you close one eye, the closed eye just have some random, very low spontaneous activity. And that spontaneous activity is not going to be correlated with the output of the cell, because this cortical cell is driven by the open eye. So there is no correlation between this guy, and that is disconnected. So here the important key is the correlation. Okay, so you kind of reward correlation and you punish non-correlation. And that's going to be the whole thing about spike time-independent plasticity. So the subject was dormant for like about 50 years. Nobody cared about that. Everybody was happy with the LTPs, with the tetanus and stuff like that. More or less until these guys, Sackman, do you see it in here? Yeah. Sackman and Stewart make the interesting observation. Basically, up to this moment, which is in the 1990s, people were thinking kind of the classical way that this is a neuron and these are the dendrites, and normally the dendrites collect synaptic inputs. So all of those travel through the dendrite and get integrated in the soma of the dendrite, and then the cell fire an action potential, goes to the axon here, and goes to another cell. It's kind of like the unidirectional flow of information. And that's kind of like a dogma, okay? Everybody was, neuroscientists of my age, learned with that dogma. And these guys in the 90s proved that dogma is wrong. So very often, the cells, especially this one with a very long dendrite, if you initiate an action potential in here, actually the action potential not only propagates through the axon, but also back propagate through the dendrite all the way there, okay? And those guys discover actually the dendrites also have sodium and potassium channels like the axon, but in less quantity. So they transmit the action potential, not as good as here, but substantially, okay? So you initiate an action potential and it goes both ways, okay? Not one way as people thought before, okay? And here is the way to do it. You initiate you put several pipettes to recording electrodes, recording here and here and here and here. So you're stimulating here, you produce the action potential, which is this. You record it in the axon, which is this one, but then you record far away in the dendrite, okay? And here you don't record anymore. So it can travel backwards. And that was people started to think, oh, maybe this action potential going backward in the dendrite to where the action of the synapse is happening, maybe it's important for LTP, okay? And one of those guys in that time made the following experiment, okay? So, and it was cool. So here you have a pyramidal neuron the guys fill it up, the pyramidal neuron, with a fluorescent dye so they can see it. Okay, so this is the soma, and this is the dendrite. Okay, and here they put another small stimulating electrode to activate synapses only in here, very local. Okay, so very local. And then they also, with this electrode, they can produce action potentials. So here are the action potentials produced, for instance. Okay, and they can also stimulate locally in here to produce local synaptic responses. So what they did is that they pair this and this. So they give all of these bursts of action potentials, I think like force in this little burst. Okay, and this is the synaptic stimulation. So they can do it simultaneously, or they can do it out of phase. They can deliver, for instance, all of the action potentials first, and then they deliver all of the synaptic stimulation later. So when they do it unpaired, one or the other, nothing happens. But when they do both of them, you get LTP. So this is the size of the response, nothing here, and then you get LTP. So if you make, and this is interesting, so if you make the cell to fire action potentials in here, you can potentiate the responses that you're stimulating in here. Okay, so the idea is that action potentials are going that way. Okay, so if that is true, we're going to do now another experiment, a little bit of refinement. Now this is a cool experiment. So what you're doing here is the same cell, with all of these things, and you're stimulating in here, and it's a little bit more complicated, and I'm going to make it simpler. So you're stimulating here. Okay, and you record the synapses that is activating here, also in here. Okay, and then what you do is the following. So you stimulate and record, and also you are recording the amount of calcium that enter into these different places, because anytime you activate the synapse, you're going to get a calcium signal because of the NMDA receptors. Okay, remember that. And also anytime you have an action potential, you're also going to get a calcium signal because you activate some calcium channels. So the calcium signal is in here, okay? And you have two things. And so you for instance, in here, you activate, you get all of these things, and you're pairing, actually this time, the action potentials with the synaptic stimulation, okay? So you get these kind of things, all right? And you get these kind of things in there. But now you're going to do the following. You're going to put TTX in the middle. Okay, and this is imaging the calcium here, here, and here. TTX, what it does is block the action potentials because it blocks the sodium channels. So the idea would be if you put TTX locally in here, the action potentials are going to be initiated in here. They're going to travel up to there, and that's it. Okay, they're not going to go there. And what happened with the calcium signal? Well, you have a strong calcium signal in here, you have strong calcium signal in here, but here you don't get any more calcium signal, because somehow the action potential not propagated into there does not allow the synapses to fully engage into this calcium business. Okay, you follow that? And of course, what happens if you put TTX in here, and then you pair firing action potentials with the synaptic stimulation, you don't get any LTP. Now, what you do is you wait a few minutes, until the TTX goes away, and you repeat it, and now you get the potentiation. So the idea is that by preventing the back propagation of the action potentials, you don't get a plasticity. You don't get LTP. All right, and here is another early observation that was crucial for this. So what you have here, they're imaging calcium. You can put a dye that fluoresces anytime you have a calcium entering into the cell. Basically, all of this is going to be calcium signals. This is when the guys, they give like a big burst of activity, and they can record. These are spines. These are synapses. Okay, and they can record calcium entering into here, and the two circumstances. Okay, so what you're doing, you're stimulating the cell in here with this electrode to fire action potentials, and you're also stimulating locally in here outside to activate the synapses. And then you record the amount of calcium that you get into those synapses in a particular one. So you activate the synapse and you get this little amount of calcium, okay? Now you activate the action potentials, and the action potential traveling here, you open local calcium channels, so you may get a little bit of calcium in there, all right? Now what you do both of them simultaneously, okay? You pair this and that, and you get a big change in calcium. And importantly, the change in calcium is larger than the linear summation of this and that. So you have a superlinear summation, you have an interaction in here, okay? So somehow the action potential is increasing the calcium coming in much more than just adding it, okay? So that was the first important thing. And here's another cute thing. It depends on the order in which you produce the action potential and the synaptic activation. So here you can give the action potentials first, okay, and then the synaptic activation, and you get a moderate amount of increase. Basically, kind of like a summation of those. But you give the synaptic activation first, and then the action potentials, then you get the big increase. So it's asymmetric in time, okay? And do you know what is the idea? Anybody have a guess? What about the NMDA receptors? All this can be explained by the NMDA receptors. Remember that the NMDA receptors, in order to be fully active, they need the cell to be depolarized to have a very relatively positive memory potential, because that can remove the magnesium that is blocking the entrance. So in this case, what happened is that the cell, you activate the synapses, the NMDA receptors bind the glutamate, but they're not fully active, okay, because the cell is not fully depolarized. But in this moment, you bring the action potentials, and now it's fully depolarized. So now the NMDA receptor can be fully active, okay? And that's when you get the big thing. That's because the NMDA receptor keep that glutamate holding for a while, okay? On the other hand, if you deliver first the action potential, you get the calcium change, and then you activate the NMDA receptor. It's like, well, so what? The action potential is already gone. So that's the NMDA receptor can explain the asymmetry. And then it's basically we are just getting ready to the HEP idea. So here is I'm going to put the HEP idea again. So this is cell A and cell B, and basically it says that if cell A fire and cell B fire after, then that synapse should be potentiated. And the tenth postulate is that if cell B fire first and cell A fire later, you should get LTD. And those things are very much like that. So this situation should give you LTP, and this situation it should give you LTD. Okay? So that if we're connecting the dots properly. Okay? And of course people connected the dots and they did it. So here is an experiment. This is an experiment in a cortex. This is a slice of the cortex, and this is a stimulating electrode, and they're recording in here from this cell. Okay? And what they're doing here is that they're stimulating, okay, so here's the synaptic response. And here, over time, varies a little bit, but it's kind of like around the same level. And at this point, they keep stimulating at the same frequency, but then they make an action potential to fire in this cell by putting a little bit of current in here. So the cell, you get a synaptic response, followed by an action potential. Synaptic response, then follow an action potential. And then they do it for a while, and they get an increase in the response. And then, so that's cool, they did the opposite experiment. And in the opposite experiment is that it's the same response, okay, but in this case the action potential happened before the synaptic response. Okay, and in that case you get depression. So what is telling this is that both cells have to fire action potentials. The first one, in this case, it will be here to release some glutamate, and the postsynaptic also to fire. And it's the timing between the presynaptic and the postsynaptic firing that it determines whether you're going to get LTP or LTB. And that's the only thing that you need to have. You don't have to give tetanus. You don't have to depolarize the cell. You just have to coordinate the firing between pre and post. Okay? And people went more on that. And actually, what you can do, and that's very beautiful, is you can systematically vary the timing between the action potential and the synaptic response. Okay? Here is before and after, and here's, okay? So as you start to change the difference, you get, consider in this case, you activate the synapse, and then you give the action potential with a delta T. If delta T is close to 0, basically, glutamate is released, the synapse get, or the NMDA get all of the glutamate, and then you give the action potential immediately. The NMDA receptors have all of them with their glutamate in there ready to get to conduct, and they do it unhappily, and they produce the monster LTP. Okay? But if you start to delay, what happens is that the NMDA eventually is going to release the glutamate. The glutamate is not going to hold into the NMDA forever. Actually, it stays there for about, like, with a decay, with a time course of 50 milliseconds, more or less. Okay? So if you do the same thing, but 50 milliseconds after, nothing is going to happen, because the NMDA receptor grabs the glutamate, but 50 milliseconds after, it threw it away, and then the action potential came and nothing happened. Okay, so that explains this. And something kind of similar may happen in this way. Okay, actually, I'm going to tell you that the exact mechanism of LTD is not totally clear. The LTP part is clear. The LTD is not that clear. Okay, but okay. So this is a summary. So what is good about this is that it's strictly based on the temporal correlations, not in the level of activity. So if the cells, the pre and postsynaptic cells are correlated, that's going to be stronger. Okay, and it makes sense. Okay, and it's just kind of like the Haven postulate. If the two guys are correlated, are participating in the same engram, on the same part, they're going to be potentiated. Okay, and this is the beauty of this, because it encodes causality. So it's kind of like a cellular basis, a synaptic basis of causality, because basically it's pre-synaptic make the postsynaptic cell fire, so to speak. That's LTP. So it's pre-cause A cause B, then you enforce that, but it's A doesn't cause B because you're after you depress. So basically you're potentiating causal synapses, so to speak. And conceptually that's very nice. And actually, if you can go and revise the literature, you will see that this thing called spike time independent plasticity, they're probably like 10, the ratio of experimental to theoretical paper is 1 to 10. So for each experimental paper, there are 10 guys analyzing the consequence of this, because it's very beautiful, basically. And you can build machines, that learn into this way. Okay. And the other thing that people like it of this is because you could record from a behaving animal. It's not that difficult to record the trains of action potentials in a given cell. You can have, actually, you can now, this time you can record from hundreds of cells. Okay, and from there, from those trains, over the timing of those trains, you could, in principle, predict if these two cells are connected, whether going to be a potentiation or depression or nothing. Okay, so you could predict, and this is information that is available for you, and you don't have to go inside the cell and measure the NMDA receptor activation and those things. So you can just see from the outside if this cell is firing and this cell is firing. Oh yes, yeah, that's going to be LTP, and very often it happens like that. So it's a much more powerful way of studying and predicting plasticity. Okay. Now, how it works? Well, I told you that for the pre and post, it's easy because this is an NMDA receptor. For the post and then pre, the LTD part, there are several hypotheses. I just put it in here. You don't have to, I mean, you can read them, but it's basically there are alternative ideas and nobody have the last saying on this, okay? So, so one, the more common one is that they say, okay, so when the action potential change, you open a little bit of calcium in there, you get, and then the NMDA is activated, so you, that little calcium pile on top of the other one, so you just kind of have like a little bit more than normal, and that's enough for LTD. And that's the most popular one. The other one, which is unpopular, but it was popular because of this endocannabinoids part, okay, the idea is that you produce some endocannabinoids in the postsynaptic cell with this paradigm, and then those act on the presynaptic cell and make everything to reduce. But it's a little bit convoluted. So just remember that this is the most popular one, and that's it. Okay. Now I'm going to go to the second part. So basically, I would say 90% of the people now will agree that the spike time-independent plasticity, in which the order of the action potentials in the pre- and postsynaptic cell code for the polarity, whether it goes up or down, and the magnitude, whether it's a lot or little, is the best model of how plasticity happen actually. And this is the thing that you have to see when you're trying to learn how you encode memories. And all of that is supported by the NMDA receptor, but actually is the order of the firing important. Okay, so let's go to the next part. So the next part is about metaplasticity, or the plasticity of the synaptic plasticity, okay? And the idea is, it came by the observation that LTP and LTD are intrinsic positive feedback mechanisms. So they tend to be, they're not stable. If you make a network, and people realize that immediately in the 80s when they start to build networks, neural networks, you know, endowed with LTP only, or LTP and LTD, but it's kind of like the initial setting was slightly to LTP. What happened is that if you have synaptic activity and the network was initially biased towards LTP, well, you're going to get LTP and you're going to get a bigger synaptic activity in the network. More synaptic activity, it will produce in turn more LTP. And more LTP, eventually, in the next round of activation, is going to produce more synaptic activity. And you have basically a loop that it will make the whole network to go saturate. And actually, network used to saturate. So they had to put all kind of addendums, you know, oh, okay, it's going to saturate, but then we're going to make it like a spontaneous decay so it doesn't saturate. But that's kind of like not very good, especially if you want to preserve a memory forever. So you cannot put decay. And the same thing happened with the LTD. So if the network is originally just by chance a little bit of speck, you know, biased toward LTD, in the next round of iteration, next time you activate the network, you're going to produce some LTDs, and you're going to have less activity, and it's more likely you're going to produce LTD next time because you need less activity for LTD, and then eventually the whole thing is going to go down. So those were very unstable networks. You know, you iterate them, and then one goes up and one goes down. All right, and the only way to make it stable, it was just to precisely make LTP equal to LTD to begin with, and that's very unbiological. So you need to have some corrective mechanism, some homeostatic mechanism. And the way that people have done that, it was actually, and again, this is one of the cases, is a theory preceding experiment. The way to do that, it was to tie synaptic response with neural activity in a way to modulate plasticity. All right? So the idea, it would be if you have a negative feedback that you have, if the neural activity is reduced, then you can increase synaptic response somehow, well, then you can correct. So you need a corrective mechanism. And the first corrective mechanism that showed up in the neuroscience and there was another experiment that prompted this guy. It's showing here? No, it's not showing here. The guy is Cooper. They invented this mechanism. Cooper, it was a physicist, and still alive, who got the Nobel Prize for the Cooper pairs in superconductivity. And after showing that, he found that there was no more interesting things in physics, and he said, I'm going to go to neuroscience, where there's still kind of interesting problems in there. So then he was going to solve the problem of the instability of the networks only with LTP and only with LTD. And he was kind of motivated by this experiment. So it's experiment kind of like the Hewell and Bissell experiment I told you before, in which you take a cat and you close the eye. Okay. So normally in the cat, it's a little cat, you stimulate one eye and you get responses. You're recording here from one cell in here in the visual cortex. Okay, you stimulate the right eye, you get responses. You stimulate the left eye, you also get responses in here. You have some spontaneous activities always. Now, after 2 days, you close this eye for 2 days, and what happened is that the open eye still responds, but the closed eye doesn't respond anymore. Okay. And you understand that very well with the spike time independent plasticity, because this guy, the closed eye was not firing, so it was uncorrelated, so it was depressed. Fine. So far, so good. Everything is working, except there was another addition to the experiment, which is called reverse suture. So you suture one eye, and what you do is you do the classical suture of the eye, but then you open that eye suture and open the other one. Okay? So what happened is that at this point, remember, this eye is not longer connected to the cortex.